Dear students, so far we have studied something about human health and diseases, then also the immunological aspects, how to control, that is alcohol, drug abuse, etc. That lesson is over. We have to pass on to the next lesson, microbes in human welfare. What is the role played by the microbes, the microorganisms? How are they beneficial to human beings? And that is the chapter, Microbes in Human Welfare. For biozoology, the chapter is 8, and for zoology, the chapter is 9. That's the difference. But the content will be the same for both biozoology and zoology. Now, what are microbes? The study of microbes is called microbiology, which includes microscopic organisms like bacteria, fungi, certain algae, unicellular algae, viruses, virions, and also prions. Prions means without any genetic code. The outer protein itself is acting as what we call the causative agent. So, they form the major components of the biological system on Earth. You know that one, some of the microbes, not all, some of the microbes are beneficial to human beings and contribute to the human welfare. So normally they are present universally, they are all called as omnipotent, present everywhere except the salt regions, salt water. So they cannot tolerate the salt water, that's why they are absent. But generally speaking, they are omnipotent, present everywhere, or cosmopolitan in distribution. They are present in soil, air, water, even within the body of living organisms like the plants and the animals, including human beings. Then, they can be cultured. The viruses cannot be cultured, you know that one, in an artificial medium. But you see, the bacteria and other microorganisms like fungi can be grown on a cultured medium to form colonies, and that colonies are visible in nature. So it is possible to culture bacteria or fungi in a cultured medium, and they can grow and multiply to form colonies. Such colonies are visible in nature. Now, the microbes are actually beneficial to human beings in different fields, either in household products or in industrial products or wastewater treatment and management or biogas production, or acting as biological controlling methods, or just like biopesticides like Bacillus thuringiensis, or they are also acting as bioremediation, cleaning the environment by biological option is brought about by some microorganisms. So, by cleaning the environment, the microorganisms are acting as scavengers. So, these are some of the actually that is things related to the microbes. Now we have to go for what are the benefits, the beneficial activities performed by these bacteria or anything else, any microorganisms towards human welfare. In this aspect, I would like to start with microbes in household products. We are getting a number of products, the dairy products, or we can say for example the dough, the one which is used for making idlis and dosa, or the one which is used for making a bread. All these are all coming under what is called household articles or the products. So now let's take the first part, microbes in household products. Okay, I'll continue. Now let us see the role of microbes in household products. In our day-to-day life, we have a number of products just we can get with the help of the microbes. For example, the fermentation of dosas, idlis, and we have the preparation of dough for making the bread or for making what is called these idlis, cheese, curd, yogurt, vinegar preparation, all dependent on mainly what we have, that is the microbes. Now let's take first one. What's the role of lactobacillus bacteria? So the lactobacillus bacteria, a group of bacteria responsible for the conversion of milk into curd and also digesting the milk protein. And the curd is more nutritious than what we have the milk. Let's see now. Now this lactobacillus bacteria, a group of bacteria, they act as probiotics. We have prebiotics and probiotics. I'll discuss later. So probiotics are nothing but living microorganisms, normally formed in the gut as a flora, concerned with restoring the gut flora and promoting the gut flora, that is protecting the gut flora. So it has beneficial effects. Living microorganisms formed in the human digestive tract, and they are constituting what is called the intestinal flora. Now, the lactobacillus bacteria include a group of what is called different bacteria, commonly called as lactobacillus bacteria or LAB. What are the bacteria? So bacteria such as what become Lactobacillus lactis, then Lactobacillus acidophilus and Streptococcus lactis. These are commonly called as lactic acid bacteria, and they are acting as probiotics, helping the human population just living in the what is called the gut and helping the growth of the flora, promoting the growth of the flora, restoring the floral content, that is in the flora means you know that from the plant materials because bacteria coming under the plant only. Now what is the role? These bacteria just now check the growth of pathogenic bacteria, those which are harmful to humans in the digestive tract, particularly the stomach. So these bacteria check the growth of pathogenic bacteria in the stomach and other parts of the digestive tract. Now we have to go for what is the role of microbes in dairy products, in preparation of dairy products. Just like what we have the curd, yogurt, just we have the cheese and also paneer. Let's see now. Under the household products, now we have a number of what is called the milk products or the dairy products are being obtained with the help of microorganisms. What are the different dairy products that are obtained by the activity of microorganisms? Okay, number one, see the role of microbes in the production of dairy products. That is the second activity. The first one, killing or checking the growth of pathogenic microbes. Now number one, the product, dairy product curd, the common product. So the conversion of milk into curd is brought about by the lactobacillus bacteria. That's why I mentioned simply lactic acid bacteria. This is called normally lactic acid bacteria because it produces a byproduct during the conversion of milk into curd or yogurt, etc. So the lactic acid bacteria act on the milk and convert it into milk, thereby digesting milk protein. So if you are taking the milk contains one, the protein casinogen or casein. The second one, the sugar, a disaccharide lactose. Now, normally when the bacteria are acting on, that is the milk, they convert the milk sugar lactose into what is called normally lactic acid by passing through glucose metabolism. And also they can have the ability of converting the milk into curd. So, LAB act on milk and convert it into milk. During that process, what's happening? Thereby digesting milk protein. The milk protein is nothing but the casein. So it is being digested. Coagulation of the protein occurs during the conversion of milk into curd. So we are adding a small amount of curd that is called as inoculum. This is otherwise called as inoculum as a starter. You know that when we are converting the milk, your mother is adding a small amount of, that is curd, into fresh milk. So there is nothing but we are adding the lactobacillus bacteria or the lactic acid bacteria. Adding just what is called curd into fresh milk is nothing but what we call the starter or inoculum in the form of curd with millions of lactobacilli. At suitable temperatures, so an example that is 40 degrees Celsius or less than 40 degrees Celsius, what is happening? The milk gets converted into a curd. This is by the activity of what is called the lactobacillus or the lactic acid bacteria. Now, if you are comparing the milk with the curd, which is more nutritious. So when you are comparing the curd is more nutritious than what is called actually the milk. The reason for that one, it contains a number of organic acids useful to the body, and also it is rich in what is called vitamins, just like what we have the fat soluble vitamins. That's why the curd is more nutritious than what we have the milk because of the rich amount of organic acids, and also that's what we have the vitamins, particularly the fat soluble vitamins A, D, E, K, like that. Okay, that's about the first actually the product that what we are getting. The next one we have to go for the yogurt. Okay, let's take that one. Now the second, the dairy product is yogurt. So it is the dairy product obtained by the bacterial fermentation on milk. Two events are happening during the process of yogurt. One, the coagulation of milk protein. Number two, the sugar lactose in the milk gets converted to lactic acid. Okay, that's the two things happening during the process of preparation of yogurt. Now, two important bacteria involved in converting or just in the fermentation of milk to form yogurt. Number one, Streptococcus thermophilus and Lactobacillus bulgaris. They have dual functions as I mentioned earlier. They convert the milk protein casein. That is normally they cause the coagulation of the milk protein. At the same time, the sugar present in the milk, what is called the lactose, gets converted into lactic acid. That's why I said dual functions. Coagulation of milk protein as well as what we call the lactose fermentation to form lactic acid. It's a kind of fermentation one. Now we have different flavors in the case of yogurt. It's having a pleasant flavor. The flavor of yogurt is due to the presence of acetaldehyde, one of the products that formed during the fermentation process. Acetaldehyde is responsible for giving a specific flavor to the yogurt. Now the third one we have to go for what is called the cheese. What is cheese? How is it prepared? What are the different types of bacteria that are involved in making cheese? Okay, we'll see. Now the third dairy product that is obtained with the help of microbes is nothing but cheese. So it is the dairy product which is normally obtained with the coagulation of milk. That is what is called the coagulation of milk protein, we can say the casein. During the formation of what is called cheese, what's happening? First the milk is acidified with the help of some what is called acids. Either in the form of vinegar, we have acetic acid because vinegar is an acidic acid. So with the help of actually an acid. So during cheese preparation, what is happening? The milk is first acidified with the help of some acids. Then the enzyme, the one which is synthesized artificially, what is called the rennet enzyme. It is nothing but the rennet what we have in the stomach secreted for the coagulation of milk that is being taken or that is being ingested. So rennet is nothing but artificially synthesized rennet enzyme. See, the human body, the stomach, it is responsible for converting the milk protein, just nothing but the curling of what is called the milk. That's why it's called the milk curling enzyme. So rennet is nothing but what is called rennet, an artificially synthesized enzyme. So it is added because it is the one which causes the coagulation of the milk. So rennet is added to coagulate the milk. So we are getting solid what is called substances. So the solid substances are separated from what we have if there is any liquid and then pressed into what is called actually cheese in the form of what is called the flakes or in the form of just what we have a sheet, etc. Now what are the bacteria that are involved, what are the microbes that are involved in the preparation of cheese? So cheese is made by starter bacteria. They are in the form of Lactococcus or Streptococcus or Lactobacillus. These are the bacteria that are involved in making the cheese. So acidification followed by the addition of enzyme rennet to coagulate the milk. That is the one we have during the process of preparation of cheese. This is another product. Now we have to go for the last product related to paneer. It's often called as a cottage cheese or fresh cheese. How is it prepared? Let's see now. The fourth product that is obtained by the effect of microbes, that is a dairy product, is paneer. It's a cottage cheese or otherwise called as a fresh cheese. So it is most commonly used in South Asia, particularly in India. What are they preparing normally? Just this one. So it is prepared by acidification. So it is prepared by the addition of what's called the lemon juice, which contains, you know, that one, citric acid or vinegar, that is acetic acid, or any other acids to milk. So it causes the coagulation of milk and separating the water. That's the main purpose of acidification. So it is prepared by the addition of lemon juice or vinegar or other acids to milk. Now, in the case of different types of what we have paneer, in the case of Swiss cheese, a kind of paneer, we have large holes of present. The large holes present in these cheese are because of more amount or more production of carbon dioxide that causes porosity in the cheese or in paneer. This is caused by a bacterium. The name of the bacterium is called Propionibacterium shermani. Propionibacterium shermani. This is the bacterium responsible for making large holes in the Swiss cheese, a kind of paneer, and there you have what is called carbon dioxide is available more and more, that result in porosity. So that's about the paneer, just cottage cheese or fresh cheese, commonly used in India as well as in South Asian countries. Okay, with this I conclude about what we have the role of microbes in the production of dairy products. Now we have the next one. What is the role of microbes in making? That is what is called in fermentation of what is called the idlis, or fermentation of the dough to form idlis or dosas, or fermentation of dough to form what is called actually the bread. Okay, that's the next one. Either for that is making idlis or dosas, or for baking the bread, fermentation is needed. So the dough should be fermented. Now in the case of making idlis and dosas, the dough should be fermented. So dough used in the preparation of idlis and dosas are fermented by a bacterium. The name of the bacterium, Leuconostoc mesembryoides. That's the one responsible for the fermentation of the dough used for the preparation of idlis and dosas. But for making bread, the dough that is used is being fermented by a different, that is actually organism. That organism is nothing but what we call the unicellular fungus, the yeast. Baker's yeast is used, not the bacteria. So the dough used in the preparation of bread is fermented by yeast. Saccharomyces cerevisiae, commonly called as the baker's yeast. Baker's yeast for making the dough to be leavened. Now what is happening? So in the bread you see that one, it contains carbohydrate. It's being broken to form glucose. So when glucose present in the dough is fermented. Now the fermentation of just glucose in the absence of what's called oxygen. Normally fermentation is an example for anaerobic respiration. Okay, here fermentation occurs in the absence of oxygen. So in the absence of oxygen, what will happen? The carbohydrate present in the dough is broken down into glucose. Now the glucose is fermented to form carbon dioxide and large amount of ethyl alcohol. And the formation of carbon dioxide and ethyl alcohol is mainly responsible for the leavening of bread. When the leavened bread is baked, what will happen? The carbon dioxide and ethyl alcohol present in the leavened, what is called the dough, that is evaporates, resulting in porous and soft nature of bread. That's what's happening. When the leavened dough is baked, carbon dioxide and ethyl alcohol evaporates, giving porous and soft nature to bread. So the soft nature of the bread is due to the porous nature, that is because of carbon dioxide and ethyl alcohol both form due to the fermentation of glucose, thus normally being evaporated. That results in soft and porous nature of the bread. That's about what we have the role of microbes in the fermentation of dough, either the one used for making idlis or dosas or for making the bread. Now the last one under the household, that is the products. The role of microbes in household products, that is single cell protein, SCP, single cell protein. 1. The role of microbes in household products. A single cell protein, SCP. So it is referring to what? The edible unicellular microorganisms, which contains protein rich. Such organisms are called single cell protein organisms. So the edible microorganisms together call us actually that is single cell protein. So an example of unicellular alga, spirulina, you know that one, there is one which is being also taken too. That is the space by the astronauts as a food in the form of capsules. Other unicellular organisms which are used as SCP are bacteria like alkali genes, then the chlorella, another unicellular alga, then mushroom. So you see that one agaricus, mushroom agaricus. So these are all actually the structures or the plants which are being used as single cell protein. So from these microorganisms we are extracting the proteins. So these proteins are extracted either from a pure culture of only one organism or a mixed culture of, for example, yeast, bacteria, algae and fungi. And these are all actually forming ingredients or what we call substitutes of protein-rich foods. They are being added as ingredients or substitutes for protein-rich foods and are suitable for human consumption and also used as an animal feed. That's the role of SCP. This actually is a vast ocean. A number of this actually SCP structures are being produced with the help of using fermenter vessel. The fermenter vessel is the one which is used for the preparation of large mass of SCP. Okay, so we can prepare SCP either from alga or from yeast, or so Saccharomyces is also used for preparation of single cell protein. I will mention that alkali genes bacteria, chlorella alga, mushroom, the agaricus, and the yeast, Saccharomyces cerevisiae, all being used in the preparation of single cell protein, a mass of proteins as substitute for what is called actually protein-rich foods, supplemented diet, useful for human consumption, suitable for human consumption, and also for animals as a feed. So that's about the microbes in household products, their role, the different products what we obtain. We have to go for the next one. What is the role played by? That is microbes in, that is what we have industrial products. Let's see in the next class. Till then, goodbye. Thank you.